following your instructions.
All right, so I welcome everyone to the next TCBG seminar, Theoretical and Computational Biophysics seminar. Today's speaker is one of us, J.C. Gumbart, Professor J.C. Gumbart from Georgia Tech School of Physics, who was actually a former group member, a student and postdoc in the group, and still works very closely with our group. Again, before I introduce the speaker, I would encourage everybody to keep their cameras on, but please turn off your microphone until the time for questions and answers. And if there are burning questions, you can actually paste it in the chat box so we can interrupt JC. I hope he doesn't mind to be interrupted for burning questions in the middle of the talk. Yes, okay, so now this is the formal part of the introduction. JC received actually his bachelor's degree in physics and mathematics from Western Illinois University in Macomb, Illinois, and then his master's and PhD in physics from University of Illinois Urbana-Champaign, and he did his PhD under supervision of Klaus Schulten. After finishing his PhD here, where he was very productive actually, he did also a couple of papers with me as a side project during that time. So he continued actually after a year of postdoc in Beckman, so he moved on and received the prestigious Director's Postdoctoral Fellowship from Argonne National Lab, where he actually worked with Benoit Roux for a couple of years before joining the faculty of the School of Physics as an assistant professor at Georgia Tech. He was soon promoted to associate professor at the School of Physics and currently also has adjunct faculty positions in chemistry, biochemistry, and the School of Biology. So he has received quite a few remarkable honors and awards, including the NSF Career and a K22 Career Transition Award from NIH, as well as the Director's Postdoctoral Fellowship at Argonne National Lab that I pointed out. Research-wise, many of us know actually JC and his sort of systems and this interest. So he continues to be highly interested in membranes and membrane proteins, especially outer membrane systems, but at the same time also has a great interest in developing software tools. Many of us know actually FFTK, which was a great collaboration between JC's group and our group in Urbana, and very useful, and he continues actually to work on that tool. So he had a long list of publications. I don't want to go through of them, but now today we're going to hear about some new systems in his group that you can see on the screen. So namely simulations of COVID or SARS coronavirus 2 proteins. Thank you very much, JC, for coming back. So we look forward to your seminar.
Yeah, so thanks, Ahmad, for the wonderful introduction and for virtually hosting me for this seminar. So it's really a nice opportunity for me to talk about some of the work we've been doing for the last now 8 months, like many of you as well, looking at proteins in the coronaviruses. Okay, so what you see here is the spike proteins. I'll talk more about that in a bit. So normally what I would be talking about is gram-negative bacteria like E. coli here, and proteins, particularly in this outer membrane of gram-negative bacteria. So for example, some of the projects we've done in that area, looking at antibody binding to proteins in Salmonella, or secretion of toxins across the outer membrane in this case CDIB. So that was a collaboration with Susan Buchanan, who Ahmad knows quite well as well. Or very recently, some work we've done on even more unusual bacteria like Coxiella and how they anchor their cell wall to the outer membrane. And so I would love the opportunity to come back at some point in the future and talk about these projects, once everything, once our current state is over. But for the time being, I'll talk about our work on coronavirus proteins. So most people, I think, by now have some familiarity with this. So I'll just briefly go over the quote unquote life cycle of the virus. So first, it binds to a receptor on a human cell protein or on a human cell and gets endocytosed, where it releases its genetic material, in this case RNA. That RNA gets replicated in order to produce new viruses, and also translated and trafficked in the cell and processed. And then finally, they assemble the new RNA and proteins, and then the viral particles are released. So what I want to focus on today are a couple steps in this process, namely, first, the translation step and the processing, and then also the initial binding step of the spike to its receptor. Okay, so talking about the translation and protein processing step. So it's actually pretty interesting that many of the proteins of the SARS-CoV-2 virus are initially produced as a so-called polyprotein. So rather than producing one at a time, a bunch of them are in sequence together. And so in order to break these up, the virus also encodes two proteases to cleave this polyprotein at specific sites named MPRO or main protease and PLPRO. So the main protease usually comes as a dimer, looks something like this. And the active site you can see is labeled there with cysteine 145 and histidine 41. So two active sites in the dimer. And if we zoom in on that, we can see, so again, you see the cysteine 145 and histidine 41, but there are also a number of other histidines around the active site. And so if you've if you've ever run an MD simulation in your life, you have come to deal with the fact that crystal structures typically do not have hydrogens present. And so we need a way to determine what the protonation states of these various histidines are. So while oftentimes we might go to some software or even just guess, at least back when I was doing it initially, we need a smarter way here. And so what we came up with in the lab was to use MD simulations to test the stability of different combinations of protonation states of these histidines. Okay. So we simulated 12 different combinations of protonation states for the MPRO dimer. Modulating the protonation of the histidines, either, for example, HSE or HSD, meaning that we put the proton on the epsilon nitrogen or the delta nitrogen, sometimes even on both. So if we think the histidine may be in a protonated state. And then we also modulated the catalytic cysteine 145, which we do know has to become deprotonated at some point in order to carry out a nucleophilic attack on the substrate. So we simulated these different combinations for 20 nanoseconds each times 3 copies. And in addition to the apo state alone, we also looked at it with 2 different inhibitors bound that had already been crystallized. And so here's what we see. So just a couple examples. The one on the left and on the right, you can see they only differ by the protonation of 2 histidines, that is just flipped in one versus the other. And we can see in the one on the right that the inhibitor actually is very unstable and comes unbound, whereas the one on the left is stable. And so we think this is a good way of judging that the protonation states chosen for the one on the left are correct and the one on the right are incorrect. One of the things we realized in doing these various simulations was that the protonation states are not uniform and in fact do vary, whether it's the apo state or or different inhibitors are bound. And so we think it's really important to consider this. And in fact, our paper on this has recently been accepted in chemical science. So this one here, inhibitor binding influences the protonation states of histidines in SARS-CoV-2 main protease. And this was work done with our colleagues at Oak Ridge National Lab. You might recognize one there, for example, Josh Vermas, and also some collaborators at Novartis, where they're also very interested in how to determine the protonation states of these proteins. Okay. So that's the main protease. There we go. I want to move now to the spike protein, the one that most people are familiar with, or at least have seen. So this is so named because it forms this characteristic corona on the surface of the viral particle. You can see there in the cryo-EM. And in fact, I believe you had Gerhard Hummer speak to you maybe a month or two ago. This paper I cite there was, he was also involved in that work. Oh, well. Okay. So the spike protein is a trimer of this S protein attached to the viral membrane envelope. So I show it there on the right. Just to note, we do not include the membrane bound part. We only focus on the part above the membrane. So one thing to emphasize, it's not just a protein, it's really a glycoprotein. So this glyco references, okay, I'm sorry, you're gonna see my Slack. I should have, oh, well, maybe. Okay, hold on, I'm sorry. Let me turn this off. Okay, let me go back. Okay, so and let me hide that. So the glycan is references a sugar. So there's many polysaccharides dotting the surface of this protein, and it's not just a viral property. It's actually a property of many human proteins as well. About half of them are glycoproteins. So it's something that, at least I know in my experience, we didn't pay as much attention to as we maybe should have in modeling the behavior of proteins. Okay, so this process of glycosylation it's actually a post-translational modification. So it happens in the endoplasmic reticulum and in the Golgi apparatus of the host cell. And just for reference, so there are a lot of structures of spike protein going around or that have been solved. We're using, for the closed state, we're using one of the later structures, PDB code 6XR8. We're using that one in particular because it resolves some features that were missing in previous structures. For the open state that I'll show in a bit, we're using an earlier one, 6VYB.
So JC, can I ask a quick question? Did any of the X-ray structures include all the glycans?
No, that's a very good question. So they only often include maybe one at a given site, just because they're so flexible.
And people assume they're different sugars. So who said which ones do you use where?
I'll show that in just a moment.
Okay, thank you.
Yeah, sure thing, Sam. Okay, so one thing to note, so a lot of attention has been drawn recently to these glycans, but coronaviruses are not unique in this. There's many viruses are glycosylated around the surface of their proteins. And in fact, coronaviruses are not the most glycosylated. There are relatively fewer glycans or glycosylation sites than many other viruses like HIV, for example. They can be part of what an antibody recognizes, the so-called epitope. So just for reference, but because they're so variable, it's not as good a site for an antibody to recognize because it can more easily be mutated away from. Okay. So for preparing our systems, we had to go from these structures, these cryo-EM or X-ray structures in which there are very few glycans present. Like I said, maybe just one per glycosylation site because the glycans are relatively flexible, and so therefore they get washed out essentially in the averaging. So to go to this, we had to do a in silico glycosylation. And to make the decision of what to put at each site, we looked to a lot of the mass spectrometry data, where indeed they find that there are multiple glycans present for a given site, or what I should say, or there's different probabilities of glycans at each site. And so we typically made our choice based on either looking at synthesizing from multiple mass spectrometry studies or picking the one that was just most prevalent at a given site. Although to be clear, there's you can imagine there's a large number of combinations possible. And so at the end, we're selecting just one. Okay. So this is kind of what it looks like. So just as we have to use cartoon representations for proteins, we're doing the same for the glycans. So you can see the difference in the cartoon where the different shape represents a different type of shape and color represents a different type of glycan. And then below, just so you can have some sense, are the actual atomistic depictions of the various glycans. So in preparing the system, so we figure out how to add the glycans and which ones to add. We also had even though I said we used a structure that was more complete than previous ones, we still did have to add some missing loops and atoms. And then of course, solvate it and add ions to make it a more realistic system. And so again, to remind you, this is without the membrane part, but it's still already about 700,000 atoms. Okay, so we wanted to
interrupt with a quick question, JC.
Yeah. Yeah, sure, Jerome.
Thank you. Do you know if there is any glycosylation present on the spike ACE2 interface?
There is, and I will come to that at the end of the talk.
Yes.
Yeah, that's definitely something we considered though. Okay, so one of the things we wanted to address with these models is to try to understand and quantify the opening, the closed to open transition. So here you see the closed state. Here is the open state. Let me see, can I move this further? No. Okay. So in order to bind its receptor on the human cell, it has to open up, so which means one domain of one of the S proteins in the trimer has to become exposed. And so it looks something like this when it binds. So there's a portion of the human receptor protein ACE2. Again, I'm not showing the part of it that actually anchors it to the membrane. Okay. So one of the first questions we asked or sought to answer was, what is the free energy difference and pathway for this opening transition? Because if we can understand that, we can also understand perhaps the role of different mutations, the role of the glycans in modulating this transition and so on. Okay. So to obtain a free energy profile, we have to select some coordinates that we think are relevant to the transition. So we focused on trying to find two coordinates in this case. And if we were to just run an unbiased simulation, we would be stuck in one state or the other typically, because we just can't run long enough to sample high energy states, as is usually the case. So for example, if we started from the closed state, and in fact we have run microsecond simulations, it never leaves the closed state in our simulations. So instead, we want to use, we turn to a method called umbrella sampling, where we, in order to better sample our space, we populate a grid in this case, two-dimensional space, and restrain a system at each point within the grid. So something like this, thereby enforcing uniform sampling for every grid point on our free energy surface. So not only that, we used a variant of umbrella sampling called replica-exchange umbrella sampling, where not only do we fill this space and restrain each system to a grid point, we allow them to exchange with one another in order to further accelerate the sampling and ultimately convergence of the free energy calculation.
So what are the reaction coordinates, JC? Next slide.
Yes, exactly.
Okay.
That's about to come.
Yeah, okay.
Well, or it will come in just in a couple minutes. So first, I want to emphasize, though, that the amount of computational resources required for this calculation become quite tremendous. We're doing this in 2 dimensions. It's a 700,000 atom system, and we have hundreds of copies of it that have to be run simultaneously in replica exchange umbrella sampling. So to do that, we were fortunate enough to get an allocation of time at Summit, or on Summit at Oak Ridge National Lab. So I think many of you have some familiarity with Summit. It was first constructed a couple of years ago and was recognized last year as the world's fastest supercomputer. So ranked #1 in the top 500. Well, okay, so as of this summer, it is now the nation's fastest supercomputer and the world's second fastest. It was dethroned by a new one just built in Japan. And just for some reference, it has a huge number of millions of cores spread over 4,000 nodes, 27,000 of these V100 GPUs, and it cost $200 million to build. So we're very lucky to get the opportunity to run on this. Okay, so figuring out our collective variables. So first, we defined a number of different domains within the spike. You can see here. There we go. So like N-terminal domain, C-terminal domain 1, 2, and 3. So the arm being the part that has to lift up in order to open and expose the binding site. And we initially considered a number of different collective variables for biasing in our calculation. So you can see here some different angles, distances, and so on. And so we started out by using steered molecular dynamics to try to see which ones or which combinations of these were good at describing the transition from the closed to open state. Ultimately, we selected 2, as I mentioned. So one being a distance of this arm that has to raise up from a stable domain in one of the other proteins in the trimer, and then also a dihedral angle between four different domains. So these are our 2 collective variables that we're biasing that we believe are representative or good at describing the transition from the closed to open state. So with these 2 in mind and filling our 2-dimensional space for replica exchange umbrella sampling, so we had 330 replicas, and we ran them for up to 48 nanoseconds per replica, giving us a total for one free energy calculation, a total of up to almost 16 microseconds of sampling of the spike protein. So if you were to do this on a single PC, it would take you on the order of 5,000 years to do this calculation. Of course, you couldn't actually do that, but for for point of reference. Maybe a better unit that we're more familiar with is about 1,000 grad students, PhDs. So still completely impractical or impossible. And if you tried, you would just be waiting, you'd get very frustrated, and eventually the calculation would outlast you. But with Summit, we can do this calculation in 3 days. So that's what a $200 million supercomputer gets you. Okay. All right, so here's an example of a two-dimensional potential of mean force. So again, along our two collective variables, our dihedral angle phi and our distance for this arm domain d. So you can see the closed state is in the lower left and the open state is in the upper right. And so we piece together the different states. And so you can see here kind of a movie of how it opens along this least free energy pathway connecting the closed to open states. Okay, so I know looking for myself at least, you can get some idea from looking at a 2D potential of mean force, but I don't always get a sense of the numbers so precisely. So we like to project this onto a 1D pathway. And you can see there, so this least free energy pathway here looks something like this. So again, going from closed on the left to open on the right. So we can see that the barrier to opening in this case was 13 kcal per mole, or about 22 kT. And the difference in free energy between the in states is about 1 kcal per mole. So this, we don't have a lot of experimental data to compare against, but at least from populations in cryo-EM images, they think for this variant that we simulated, they're about equal populations of closed and open. And so that would tend to suggest that the free energy difference between the open and closed is pretty minimal. And so at least we feel pretty good about the number that we got, that it's also pretty similar between the open and closed states.
JC, can I ask you a question?
Yes, please.
So to see the points in your grid, so do you start from both structures, so from the open structure of the cryo-EM and the closed structure?
That's a great question. So we did start from both the closed and open to seed the states in between, and we used metadynamics to start filling out the space.
Okay, thanks.
Yeah.
Yeah, JC, that's Zan. So I'm sort of wondering, why didn't you just use well-tempered metadynamics? It gives you, that's the only method I've used that ever agreed with within a kcal or two of an experiment.
So at least in my hands, we've had issues getting metadynamics to converge. So we are using well-tempered metadynamics, at least to fill out the space initially. But at least just in my own experience, I've had better luck with umbrella sampling. And so that's why we then moved to that. Okay. But I think it's just more that we just don't have as much practice with metadynamics. Because
it is faster, isn't it?
I don't know. I know it's thought to be faster, but again, in our hands, we've had issues with convergence in the past, and that's why we kind of moved away from it. But I don't mean to slide it, just that we're not experienced, I think, enough with it to maybe use it correctly.
We had a similar experience, actually. We had always, especially in multidimensional, it becomes very difficult to get converged sort of landscape.
Yeah, we often, at least to go into the details a little more, we often see it like just continually going back and forth between one or the other being the minimum, one side or the other.
Thanks.
Mm-hmm.
JC, I have a quick question. So then this result implies that this is more like a pre-equilibrium and not as much of an induced fit. Is that correct?
Sorry, can you
So
repeat?
this like, let's say a 1 kilocals per mole mole difference,
Mhm.
1.7 1 kilocal. So
Yeah.
then this basically it's a pre-equilibrium between the open and the closed state, rather than an induced fit of the AC2 coming in and impose.
Yes. Yeah. Exactly. I think, and that's what they tend to see experimentally, at least in the cryo-EM images, that some fraction of them are often in the open state already, and it's not that it only opens upon interaction with ACE2.
Mhm.
Right.
Okay.
Mhm. Okay. So. One thing I haven't shown anything about the glycans. So we spent a lot of effort in putting all the glycans on the protein. So I want to just highlight them here. So now you see all of them present, and I'm going to focus on two in particular. This one's at N234 and N343, so you can see how they're colored. And in the closed state, we think they're forming some interactions that may help maintain that closed state that are then disrupted in the open state. And so that may actually be contributing to that large barrier that we saw of about 22 KT for going from closed to open. And then when it's in the open state, one of these glycans actually starts to interfere by inserting itself inside the binding site that was vacated by the protein. And so this is something also that's been seen by others, like maybe you've seen in Romi's work, for example. So these glycans, they can really get in the way of things, basically. Okay, so what about if we remove them? So that was one of the questions we wanted to ask. If we get rid of all the glycans, how does that alter the free energy? So here again, we see the 2D still closed on the lower left and open on the right. And then we plot that on a 1D, or we make a 1D plot along that least free energy pathway. And we see that the barrier is significantly reduced. So now it went from about 22 KT to about 7 KT without the glycans present. The difference between the states, so keep in mind before it was 1 kcal per mole, here it's minus 1 kcal per mole, but that's still, I would say, suggesting that the two states are roughly in close populations to one another. So the only difference is without the glycans, you change the barrier significantly.
So you have a new minimum now.
Yeah, and the minimum actually does shift a little bit to the open state, but it's still all, I would say, this is all less within 1 kcal per mole. That's considered usually, within chemical accuracy at that point.
Right.
Mm-hmm.
Hey, JC.
Yes.
This is Alec. Hi.
Hey.
What force field did you use for glycans?
So we use Charm 36 for everything.
Was Alcox NB fixes?
No, we included the MB fixes that are part of Charm 36. Yeah.
Oh. I mean it's done, but have a look at his. He has this custom MB fixes that prevents aggregation of sugars.
Sorry, whose did you mention?
Adrian Alcock.
Oh yeah. Okay, sure.
He has NB fixes specifically for sugar protein interactions that prevent aggregation.
No, that's great. We actually we do still have some summit time, so that's something we can look at in the next few weeks. So thanks for that. Okay. Yeah, so that might lower that 22 KT barrier we saw. That might lower it some, although I don't know if it would be enough to lower it to 7 KT. So I still think we're seeing a difference in the glycans when they're present versus not. Okay. Okay, so what about D614G? So that was perhaps the most, well, I don't want to say, well, in a way, the most popular mutant that was that came about in the early summer. It was recognized as it quickly grew to take over by June. Basically, all of the coronavirus circulating was this D614G mutant. And so we wanted to also ask, well, what is it maybe doing? And so you see a number of news stories written about it. Okay. So it's a residue here near the center of the trimer. This is one of the reasons actually I want to emphasize that we use this newer structure 6XR8 because in the previous one it was not resolved there. So now we see in this structure that it forms a salt bridge actually with a lysine K854. Okay. So this is D614 in the original version of SARS-CoV-2 S protein. Okay. So here's what we saw when we actually did the free energy calculation for this D614G. And just to avoid complications, we did it without the glycans present. So we're comparing to the previous case with no glycans. So something interesting, so the barrier went up again. So we need to actually investigate this more to confirm that this is actually real and not an artifact or that it's confirm that it's converged, but it actually went up even higher than it was before with glycans, about, in this case, about 28 KT. But at least some comfort that the difference between the states is still relatively small. Although, when we look to experiments, they found that, in fact, actually, perhaps this one should be biased towards the open state. So we're still a little unsure how reliable this result is, and we are investigating it further. One thing I do want to note, though, so we often look inward when we have a disagreement. We look at the simulations and say, what did we do wrong? But I think it is also good to look at the experiments and say, well, okay, what did they actually measure? And one thing to note, so in practically all the experiments that you'll see, unless they were using, live virus, they are using a different version of the S protein than the actual virus has. So they reference here in this paper, for example, they use the widely used soluble version of the S protein that differs from the native one in a few ways. For example, they removed the furin cleavage site, and they also introduced this stabilizing diproline mutation. This is actually all the vaccines, or all the mRNA vaccines, are using this same version of the protein, this diproline stabilized one. And so we want to be careful to make sure we actually are comparing the same version of the protein on our simulations as they're using the experiments. So we're now repeating our calculations with this diproline mutant. So as to whether, so we still haven't answered really addressed much the question of, is D614G more infectious? And, we see, in fact, the literature, it's still pretty controversial. So we'll see a paper just a couple of months ago, evidence that it increases infectivity, but then even just a few days ago, a paper saying in Nature Communications saying there's no evidence for increased transmissibility. And so this kind of leaves me in the, I don't know right now stage. But definitely something we're still investigating.
So just a quick question.
Yes.
The percentages that you showed, is it out of the raw single particle images, or is it after the ones that are processed? Because there, of course, things get kind of thrown off, and after that, a percentage is made. So if it's possibly what you want, what needs to know is from the raw data, which you might not get.
That is a great question. And I will have to, this is again, we're reading experimental method sections better, more closely than we ever have before. So that's something we'll definitely take a look at, because I don't know for sure which they used to get those numbers.
Yeah.
Thanks. Okay, so I just have a couple more things I want to talk about. So related now to the binding of the spike protein to its receptor, ACE2. So in, I think it was in March already, there was a crystal structure, March or April, of the complex between the receptor binding domain. So that's just part of the spike protein. In fact, that domain that has to lift up for it to open, bound to ACE2, you see here in a dimer, and then you can see how it's anchored to the membrane actually by interacting with another protein. So we're going to focus just on the interface between the soluble part of ACE2 and the receptor binding domain of the spike protein. So it looks something like this. One of the very interesting things about this is that it's the exact same binding site for SARS-CoV-2 as it is for SARS-CoV-1. So same orientation and everything. So that presents a natural set of questions. You know, is binding different, and how is it different between these two viruses to the same receptor? Okay. So the majority of differences in sequences in the sequences between SARS-CoV-1 and CoV-2 are localized at this binding interface. So even though the overall receptor binding domain is 74% identical, it's only 50% identical at the interface. But as far as how they actually bind, they're actually pretty similar. So the SARS-CoV-2 does bind a little bit more strongly to ACE2 than SARS-CoV-1, but not, at least I wouldn't say it's dramatically so. Okay. So to try to get at this a little more, again, we turn to molecular dynamics. We just ran equilibrium simulations of these complexes, one for SARS-CoV-1 and one for SARS-CoV-2. Actually, we ran it each one twice.
And sorry to interrupt, but
Yes.
did you include the glycosylation on this one simulations you're showing now?
In this one, no, but in the next ones I'll show yes. Okay. Or at least in this one, it's very minimal. I think we kept the ones that were crystallized, but nothing more than that. Okay. So watching these simulations, you know, can you see the differences? Can you figure out what distinguishes SARS-CoV-1 and CoV-2? I can't. So I would be surprised if you could as well. And so we decided to ask, okay, can we, you know, we have these very smart computers. Can we ask them to tell us where to look? Okay. So we used a couple of different machine learning approaches, and specifically, we asked these machine learning algorithms to classify different frames of the trajectories, these 2 × 2 microseconds trajectories, as being either coming from a SARS-CoV-1 RBD simulation or SARS-CoV-2 simulation. So as input features, we took inverse pairwise distances for all heavy atoms within 15 angstroms of each other at the interface. So this, to be clear, we did not invent this approach or pioneer it. We're actually following the wonderful example presented by Lucy Delamate in a biophysical journal paper earlier this year. And for anybody wanting to use machine learning for molecular dynamics simulations, I encourage you to take a look at the many examples that she shares there. So with this criterion, this 15 angstroms, we ended up with about 4,000 features. So that means, in other words, for every frame of the simulation, we have 4,000 inverse pairwise distances. So we used 2 different approaches. So one actually very trivial. You wouldn't even normally think about this as machine learning, but technically it qualifies. We used a logistic regression approach. And then we also used a random forest approach. So that's one maybe more commonly recognized as a machine learning approach. So both of these, the goals of these algorithms is to try to, again, distinguish or classify frames of the trajectory as coming from SARS-CoV-1 or CoV-2. Okay. So the algorithms, they produce a ranked list of the importance of each feature. So each pairwise inverse distance as importance, meaning in its ability to discriminate between one SARS-CoV-1 simulations or SARS-CoV-2. So we summed to get it for each residue, we summed over all of them and then normalized. And so I plot here, I'm using the numbering from SARS-CoV-2, but just to be clear, it has an analogous residue in SARS-CoV-1. And we see that the 2 algorithms we used, logistic regression and random forest, have a pretty good agreement in terms of which ones they say are important. Oh. Right, and so again, just to emphasize, the ones that are of high importance means that these ones are very different or behave differently in the SARS-CoV-1 versus SARS-CoV-2. And so just to point out, this is a collaboration actually with Jerry Parks and Zhongyu Mo at Oak Ridge National Lab. Okay, so picking out a few of those that were ranked highly. So for example, 417 is a lysine in SARS-CoV-2 that was ranked the highest. And so when we look at our simulations more closely, we see that, okay, in SARS-CoV-2, it forms a salt bridge with an aspartate in ACE2, whereas in SARS-CoV-1, it's actually a valine. The sequence is shifted a little bit, so it's valine 404, which obviously does not form in any strong interaction. So this one shows that the SARS-CoV-2 apparently is better, at least for interacting with ACE2. Similarly, we see another one that was picked out as high importance was leucine 455 and phenylalanine 456. These form a hydrophobic interaction with a residue on ACE2, and we see that is mostly lost in SARS-CoV-1. Just to contrast that, as another example, asparagine 439 in SARS-CoV-2 doesn't form an interaction, but in SARS-CoV-1, it actually forms a salt bridge with a glutamate in ACE2. Okay, so we see, so the machine learning drew our attention essentially to very specific interactions that are distinct in one versus the other. So to try to quantify this better, we turn to free energy perturbation. So this is a collaboration with Chris Chapeau, who you all know very well, and I believe is watching right now. So I mentioned the experimental results. So just to put that in terms of a free energy, the difference is pretty small, as I mentioned. So SARS-CoV-2 binds about 1.1 kcal per mole better than SARS-CoV-1 to ACE2. So we picked a number of those important residues identified from the machine learning algorithms. I show here the mutations. So the one on the left is the CoV2. So the K417V means it's lysine in CoV2 and it's valine in CoV1. And the plus or minus on the free energy. So if it's positive, it means that the SARS-CoV-2 residue is better for binding. And if it's negative, the SARS-CoV-1 residue is better for binding. And we see a mix here. So I think that's an important result that there's no particular optimization of the residues in SARS-CoV-2. Some are better, some are worse than CoV-1. Okay, these results agree pretty well with experiments, thankfully. Just to highlight a couple of them, so like this asparagine 439, we see as negative, meaning that CoV-1 is better. And indeed, that's what the experiments reveal, that when you mutate it in CoV-2, you get better binding. Contrast that with, for example, this leucine 455, whereas if you mutate that one to its CoV-1 counterpart, you get worse binding. Okay. So now we put a quantitative spin on this, though. Okay, so as Jerome keeps asking and is desperate to find out, what are the glycans doing? I spent a lot of time talking about them, but I haven't said anything in this. So to understand that, we again had to turn to this mass spec data, but this time for ACE2. So the human protein, as I said, like roughly half of all human proteins are glycosylated. ACE2 is no exception. And again, here's some cartoon depictions of what we selected for the glycan at each site. Although again, to emphasize, there's some diversity here, as you can see in the chart on the left. Okay. So here's what it looks like. So again, using these very nice cartoon representations of the glycans, you can see the differences between the SARS-CoV-1 and SARS-CoV-2. So the ACE2, to be clear, is the same in both, but the receptor binding domain on top is a little bit different. So the one on the left, you can see, has 2 glycans, and the one on the right only has 1. Okay, so this glycan is actually lost in SARS-CoV-2. Okay, so again, we ran simulations, so a couple microseconds for each. So you can see they start out looking similar, but when we look at the behavior of the glycans mapped out over the course of the simulation, we can start to see some differences. In particular, this one that was lost in SARS-CoV-2, you can see now they're present in SARS-CoV-1 and is shifting some of the interactions with ACE2 glycans, or at least some of the behavior of the ACE2 glycans. So we quantified that. So it looks something like this. So just to draw your attention, so the blue is the ACE2 and 90 glycan. So that one's similar in both. And so when we look, we see, okay, in both SARS-CoV-1 and CoV-2, it's forming some interactions with the receptor binding domain. But there you can see in red is quite a difference for this ACE2 and 322 glycan. Whereas in SARS-CoV-2, it's forming a lot of interactions, but in CoV-1, it's blocked by that SARS-CoV-1 glycan that is lost in CoV-2. Okay. So we see, in fact, comparing the behavior of these two, so the N90 glycan actually interrupts some of the interactions between RBD and ACE2. And so this actually agrees pretty well with experimental results where they found mutations of ACE2 that eliminate that glycosylation site actually enhance binding to the receptor binding domain. Okay. Contrast that with the N322 glycan where mutations that abolish that glycan actually reduce binding to the receptor binding domain. Okay, so wrapping up, so what did we conclude from this 8 months of and change of work? So one of the things, so thinking back to the beginning of my presentation, so looking at the main protease, so MD simulations in particular can be helpful for making some of these critical decisions, like where to protonate certain residues like histidines in particular, which is an important step in the, will be an important step in the drug discovery process. We also saw, for example, in our two-dimensional PMFs of spike opening, we saw that the glycans can potentially impede the opening, changing the energetics, the barrier. And we also see that there's a number of differences between the receptor binding domain in CoV-1 and CoV-2. But these differences actually both can, in some cases, improve binding, but also in some cases impair binding. We saw that both in terms of the amino acid sequence and also in terms of the glycans. And so maybe if there's one message to take from that, I would say that the SARS-CoV-2 is not optimized for ACE2 binding. If there was anybody out still in doubt about whether it was engineered or something. Okay, and with that, so I'll just acknowledge, so a number of people in the lab have been in my virtual Zoom lab as we all exist in now. Anna Pavlova, Diane Lynch, and Atanu Acharya. So Anna and Diane worked on MPRO. Atanu has become an expert in glycosylation, thank goodness. Andrew Pang did, a physics graduate student, did work on the free energy calculations on Summit. And Zijian Zhang, another physics graduate student, has also been helping with all of us with the machine learning aspect of the project. And then we've also been fortunate to collaborate with a number of great people, including some, like I said, you recognize, like Jeremy Smith, Josh Vermas, Jerry Parks, and so on. Okay, and so I hit my time pretty well. Actually, I'm impressed. And there's, I guess now it's time for questions. So thanks.
Well, thank you very much, JC, for a very nice collection of results on this virus. I'm sure there are lots of questions. We already asked you a lot of questions. You can, I don't know, just indicate that you want to ask a question. I'll let you go ahead. I can start. I have actually two questions. One is, so when you did these domains, motion, distance, and the hydral angles, so you are using center of mass of these bits, I guess, right? So
Yes.
was it very slow for these two very expensive collective variables to be included in NAMD? Did it just slow down the calculation too much, or?
I mean, we only use C alphas. In the end, I think we have, maybe on order of 100 at most atoms in each collective variable. So I don't think it slows down too much, but we didn't explicitly check.
I see. And then the other question, I know that actually there are questions about this famous mutant D2G, with the experiments, and I totally agree with you. In many cases, it's not a problem with the simulations. From a physical chemistry perspective, it makes sense what we see in the simulation. Something is wrong with the conditions. The conditions of the experiments don't match simulations, for example. But do you understand how within your simulation framework, do you understand how this mutation might increase the barrier by, to what was it, more than 20 kilocalorie per mole?
I do not. That's why it was a little bit of a surprise to us, but I like to present, unfiltered results. But we do need to dig in that. And one of the things we're doing actually, so thanks, Ann. So we'll see you later. We
have a quick question if you finish soon.
Oh, yeah. Well, I was just going to say we want to repeat the calculation then to just see if it holds up.
Fair enough. Go ahead.
So, we use this saliva test here at the U of I.
Yeah, we
and
do.
Paul Hagenruther gave a beautiful talk, and that's where I first learned about all the different glycans that were on there. But have you guys done enough of a study from the mass spec? Were they on different patients? Because, there was some indication that maybe your blood type would indicate the severity of the infection. And because they also have different glycans,
right,
that distinguishes A, B, and O, type. So you selected the most prominent, but have you looked and correlated that with blood type of people?
We have not. So I think, the blood type studies, they're all correlation right now, and they're kind of weak. So we haven't considered them too much until we see like a more clear mechanism, in which case then we could investigate. But the point you raise is a very good one, that even the cell type that these proteins are expressed in can affect their glycosylation patterns. And that's something, again, we have to pay very careful attention to, coming from the looking at the experimental data.
Yeah, it was beautiful mass spec work. Wonderful collaborators. Thanks a lot. Bye-bye.
Okay, questions?
So I see Karen has a question.
Oh yeah, Karen, go ahead.
Please ask.
Hi, Jason. So the question was, so what do you think drives this open to closed conformational change in spike, seeing that they have a high energetic barrier? What do you think promotes it?
I mean, I guess I didn't do a back of the envelope calculation, but I don't know, is a 20 KT barrier completely impossible to overcome? I don't know. I think maybe over enough time, it might be able to spontaneously open. At least that's what they seem to observe. There are some other things going on. One of the things we recognize from reading the literature is that roughly half of them are already cleaved at this furin cleavage site when they come out of the cell, versus needing to be cleaved by a host protease when they go to infect. So, the short answer is I'm not sure, but there's probably a lot of other weird stuff going on that we're not capturing.
Okay, just related to it, do you think, for example, opening of one RBD increases the probability of opening the other RBD, so on? Because you can also do those free energy calculations. Now there are structures with multiple RBDs open.
That's a great question. My suspicion would be yes. I think, the way they're all, I haven't looked that closely, but the way they're all tightly packed there, I could imagine freeing one would make it easier for others to come out. And if we still have enough summit time left, we may do that calculation.
James, can I ask a question related to this?
Yes.
Do you think that when two more of these would open, could they bind to more of these AC2 proteins? Did you try if they fit by size? Would it be possible? Because then it would develop some multivalency with those multiple binders, right?
Yeah, that's a great question. We did not try, but I could imagine it's possible because at least in the crystal structure with the ACE2, it showed as a dimer. So you could imagine two of the S proteins on the spike trimer opening up and binding to two ACE2s in the dimer. But I didn't look closely enough to see, but I imagine it's possible.
So then the mutation would basically lead to multivalency, to stronger multivalent binding.
It could, but the binding, yeah. How much that would actually manifest in terms of infectivity is a much more complicated question as we're finding from looking at the literature at least.
But the strength of the binding will add, right? If you have two binders, you will have stronger binding. So then immediately it would be much stronger attached.
That's true, but I guess the question would be: is that the limiting step in infection? Is it maybe the internalization that's actually the more difficult step? And that's something I don't know if I know or anybody knows.
Yeah. Anyway, great talk. Yeah.
Thanks, Petra.
So JC, another question.
Yes, Adi.
So the machine learning trick that you showed us later on, did you think of not using it before, like when you were choosing your collective variables for the umbrella sampling?
We did not, at least at the time. I think it would be very interesting to go back. I know there are people that have worked on finding better methods of choosing collective variables, more automated approaches, but we haven't considered it yet.
Yeah, the reason for that might be those collective variables would give you a surface where the barrier is lower than what you are seeing right now. And if I say the answer is yes, because I know the answer is yes. So yeah, have a look at that.
Okay, then you have to share some stuff with me.
Yeah. So yeah,
I would appreciate it. Okay.
He has a $200 million machine. He can do these things in 3 days.
We'll have it by Thursday. I'll let you know. Yeah, tell me where to look.
Great, wonderful. Well, any more questions? I mean, I see a lot of friends here. This was a very well attended seminar, by the way, and I see a lot of old friends here.
Yeah, thanks everybody.
Good, Chris included. Yeah, good. Arvind, you had a question in the middle of the talk.
Basically, I was wondering if there is an evolutionary advantage to having surface glycans. How should we understand it biologically? What's the advantage that the virus has by having glycans?
Glycosylation?
I think, I mean, at the very least, they do help evade recognition by the immune system because they can mutate more quickly, I guess. For one, they can obscure, as we've seen from others, they can obscure binding sites on the protein surface. So I imagine that's one of the advantages. There must be some disadvantages too that I'm not thinking about. Otherwise, they would probably all be covered completely in glycans. I wonder if I were to speculate, perhaps based on what we're observing, for example, the presence or absence can affect binding to the ACE2. So perhaps too many glycans may make it harder to go from one species to another. It's just one speculation I have.
Cool. Okay, well, I mean, the glycosylation is a post-translational modification actually happening in the host cell, I presume, the virus itself. And like you said, many mammalian proteins do have glycosylation. So I think it's a general feature of many proteins. Okay, any more questions? If not, thanks everyone for attending. Thank you very much, JC. It's great to see all the good friends, Jerome.
And thank you. I think it's awesome. I thank the coronavirus to be able to access the UIUC famous meetings. So that's great. You should keep it like that, Imad.
Okay, I will. I started tweeting very actively about our seminars, and so that is helping, I guess. Good. Well, wish everyone well. Stay safe. I think it's a matter of a couple of more months before we are out of the woods, as they say. So, JC, we're going to have another Zoom meeting. You have the link for the next meeting?
I do. Let me,
Okay, cool.
I'll find it. Yeah.
Bye, everyone.
Leave.